Hey everyone, welcome to the channel of Civi Forensics. I am Srishty Sinha, a volunteer at Civi Forensic, and today the topic is forensic significance of diatoms. So, this is a topic where biology meets criminal investigation. So, diatoms play a very important role in the investigation of drowning cases. So, in this presentation, I am going to introduce you to the diatoms, their morphology, their classification, principles behind diatom test, its sample collection, and different methods of diatom analysis. So, let's get into it. Introducing you all to diatoms. So diatoms are unicellular photosynthetic algae found in aquatic environment. They can be found in fresh water or marine water, brackish water, or even in moist soil. They belong to the kingdom Protista and their phylum is Bacillariophyta. The unique feature diatoms are known for is their cell wall, which is made up of silica, and these cell wall are resistant to decomposition, and these are very unique and specific to the to a particular diatom. Its forensic significance, as I have already told you all, it is used as a biological indicator in forensic drowning investigation. It leads to the conclusion, presence of diatoms leads to the conclusion whether it was an antemortem drowning or postmortem drowning. Antemortem drowning is if the person was alive while it was drowned in a water, and postmortem drowning is the person was already dead and then it was thrown into the water body. So here it is a pictorial representation of how diatoms look, and it also here states that it belongs to the class Bacillophyceae. Now let's get into the morphology of diatoms. So they are microscopic in nature. The size ranges from 2 to 500 micrometer. Each cell of diatom is enclosed with silica cell wall, which is also called frustule, and they are overlapped if with soap like structure. It is a soap case like structure. They have two halves, which is called epitheca and hypotheca. Frustule, which is the cell wall, silicon cell wall, it has distinctive pattern of pores, ridges and striae, which makes them unique, and these are used for the identification when observed under the microscope. So depending upon the symmetry, diatoms are classified into two types: centrales, which has radial symmetry, and pennales, which has bilateral symmetry. So, the cytoplasm of a diatom contains chlorophyll A, C, and fucoxanthin. These are the pigments present there, and this gives it a golden brownish color. So next we are going to discuss about this classification of diatoms. So as I discussed earlier, there are two types of diatoms: centrales and panicles. So centrales are radially symmetrical. They have mostly marine habitat. They have the chloroplast has discoid shape and large chloroplast are present there. Raphe is absent in centrales diatom, and raphe are actually some structure which is present for mobility, which and centrales are mostly static. They doesn't move, and they has radiate striation, and of course, as the name suggests, the shape of these diatoms are circular. However, pennales are the diatoms which has bilateral symmetry. They are mostly found in freshwater habitat. They has elongated chloroplast and raphe is present, and they are mostly mobile in nature. And as the name suggests, they have pinnate striations in the frustule, and they are mostly boat shaped. Here you can see the picture of a pinnate diatom and a centric diatom with different parts. They have frustule, valve, and nucleus like a normal cell. And yes, these are the structure present here. The chloroplast, as you can see in centric diatoms, are circular. Now let's get into the principle behind diatom test. So during drowning, water enters into the lungs and ruptures alveoli. And as these organisms are microscopic, they just rupture the alveoli and reach into the bloodstream. And after reaching the bloodstream, they reach distant organs such as bone marrow. Presence of diatoms in internal organs indicate antemortem drowning, because it indicates that the when the person was drowned, it was he or she was breathing, and by breathing they swallowed the water, and then it get into the lungs and then to different body parts. It can accumulate in the liver, it can accumulate in kidneys as well, but mostly we prefer bone marrow for test diatom test. And absence or mismatch of the kind of the type of atoms mostly indicates post-mortem immersion. So here is a pictorial representation of how diatoms are accumulated into the bone marrow. So the drowning happens, and then the respiration happens, the breathing happens, and by breathing, the water reaches the lungs, and then it ruptures the alveoli and reaches to different organs, including bone marrow. And when examining the bone marrow, if we find presence of diatoms, it is considered as antemortem drowning. The person was alive before it was drowned. And if there is absence of diatom, or if there is mismatch of diatom from the original source of the water the body was found in, it indicates post-mortem drowning, or there is a foul play. So in case of drowning, we have to collect sample from two places from body, whether it's lungs, liver, kidney, brain, or bone marrow. Mostly we prefer bone marrow. Either we prefer bone marrow from sternum or femur, because these are the long bones and they have enough bone marrow for investigation. And the second sample we have to collect is water body, sample of water body from which the uh, the dead body is found. We have to take a control sample, and we have to take a sample from the scene where the body was found, and then the bone marrow analysis is done. Next we are going to discuss acid digestion method. So there are several steps in it. Firstly, we have to collect the sample. We have to collect the sample from the control sample water body, and then also we have to collect get the sample from the organs like lungs, liver, kidney, brain, or bone marrow. And then the second step involves cleaning of sample. We have to remove the dirt and any contaminants present there. The third step is tissue digestion. In this step, we will add concentrated nitric acid or sulfuric acid and hit the sample gently until the tissue gets dissolved and we get a clear solution. After we get the clear solution, the fourth step is to centrifuge them. We have to spin the sample to collect the residues of diatoms frustule, which will make us help us identify the type of diatom present. And after the centrifugation, we have to rinse it with several times with distilled water to remove any traces of acid. Next, we have to prepare slide. We have to mount the drop of residues we got after the centrifugation on a glass slide for observation. And then we will observe these slides under the microscope to identify diatoms. We can do it under light microscope or scanning electron microscope. And then we will compare interpretation. The last step involves interpretation of whatever the result is found, in which we will compare the diatom species in the tissue with the water sample to confirm it was a antemortem drowning. So here is the pictorial representation of all the steps involved in acid digestion method. Firstly, we'll get the sample, we'll clean the sample, digest tissue, and then these digested tissue are centrifuged, and these centrifuged samples, the residues are washed again and again to remove all the acid traces, and then it is mounted to a slide and observed under the microscope. Next we have enzymatic digestion method. This is the second method which we can apply. In this method, the first step is, of course, the sample collection, where we collect the tissue organ and we cut it into small pieces. And the second step is homogenization, in which we add the phosphate buffer to get a uniform mixture. Third step is enzyme treatment, in which we add any proteolytic enzyme. Most here the example says proteokinase K, and we will incubate it at 37 degrees Celsius for 2 to 24 hours until the digestion happens. Then we will add enzyme inactivation. We'll add inhibitors for enzyme inactivation to stop the digestion. After which we will centrifuge the sample at 3000 to 5000 rpm for 10 to 15 minutes, and then we'll collect the supernatant layer and washing the palette with distilled water multiple times to remove the residues. And after that will prepare the slide. We'll mount the residues to the slide and then observe it under the microscope. Here's the pictorial representation of this enzymatic digestion. In, as you can see, we'll collect the sample, we will homogenize it, we will give the enzyme treatment, and then after we get it digested, we will do the enzyme inactivation. We will centrifuge it, we will wash it, and then we will prepare slide and observe it under the microscope. This is how diatom test is performed. So here we come to the conclusion of this presentation. So it concludes as that diatoms are microscopic algae with distinct silica frustules useful in forensics. The morphology and type helps us in species identification and linking it to water sources. A particular water source has a very specific kind of diatom which are not present at different place. So this helps us to identify and establish the fact whether it was a antemortem drowning or a postmortem drowning. The extraction method which are used, they preserve the structure, and we observe these structure under the microscope for investigation purpose. Analysis of diatoms provide valuable evidence in drowning cases. So here it is. uh, Thank you so much. If you like this video, like it, comment down below and share it with your friends. Have a nice day.